So cryo-electron microscopy, so usually we use cryo-EM for short. So it's a very powerful imaging technique that uses parallel electron rays to get images of biomolecules, usually proteins, at the nanoscale resolution.
This is Dr. Laurène Donati, who recently graduated with a PhD from EPFL and is now coordinating the activities in imaging at EPFL.
So the goal is to get information on the 3D structure of the proteins. And so the word cryo stands for cryogenization. So it's the fact that in that specific imaging method, so the sample is frozen in a thin layer of ice, both to withstand the vacuum condition of the microscope, but also to keep the samples in their native state.
In fact, lately, cryo-EM has become very successful at protein observation.
It's a very popular method. Actually, it's received the Nobel Prize in Chemistry in 2017, and that includes Jacques Dubochet, who is an EPFL alumni. And so it's kind of an important topic as well for Lausanne and for the Lémanique region. And we're kind of blessed at EPFL because we're going to have a cryo-EM center that's going to open actually starting from next year. So we are very excited about this also.
So why is it so important to learn about the three-dimensional structure of proteins?
Proteins, they're like very small molecules in the human body. And they perform a vast array of functions inside the human body. So most people think of proteins as the building blocks in the muscle, and that's completely right. But they also like do a variety of things, for example, like signaling a lot, hormones, that kind of thing. So it's super important to kind of like get a knowledge on what they do. And the reason it's important to understand the structure is that proteins perform function through their structure. And for example, like because they have a certain structure, they bind to certain other types of molecules, or they carry certain types of molecules. And so if you really want to get a deep knowledge of what they do in the human body, you need to get a very precise idea of their 3D structure.
Now that's for fundamental research, which often leads to unforeseen breakthroughs in the long run. But there's another short-term reason why understanding protein structure is critical.
It's also super important, for example, in the pharmacological world, because what happens is that if proteins have a defect in their structures, then they will perform, not in the right way, and that can lead to diseases. And so it's kind of important also to have a good knowledge of their structure for drug design, for example.
Well, is there a concrete example of this?
A very powerful example is the coronavirus. So one of the first things like that people did was to use cryo-EM to get an information on the 3D structure of like what we call the spike proteins. So those are proteins at the surface of the coronavirus. And by knowing like the structure of these spike proteins, then you can dream of having like specific type of drugs or like of pharmaceutical approaches that kind of neutralize the virus in some ways. So that's one key example.
Yep. Laurène's work is part of the global effort to combat the current COVID-19 pandemic. I guess this makes it a very important work these days. Unfortunately, it's not an easy work.
There are many challenges, but one key aspect of cryo-EM is that it's a let's say computational imaging or computational reliant imaging method. So you have to have algorithms that processes the data. So you cannot directly exploit the measurements. You need actually like computers and algorithms to process the data so that you can have this 3D information you're looking for.
Especially given that the data, the images that come from cryo-EM are not extremely human eye friendly, if you see what I mean, or rather if you don't see what I mean.
The data are extremely noisy, so every kind of image processing algorithm you want to apply is going to have to be super robust to kind of work. And also you have a sheer, just the sheer amount of data you collect kind of imposes constraints also on the type of algorithms that you can design.
But this is not all. Cryo-EM raises image analysis challenges that are quite specific.
People may be familiar with the CT scanner in the hospital. And so basically what a CT scan does is that you go inside, it takes 2D views all around your body, so it rotates like this. And from these 2D views that were acquired from different angles, then you can use algorithms and reconstruct the 3D structure. The kind of key aspect to understand cryo-EM, at least the specific modality called single particle cryo-EM that we are mostly interested in, is to understand that I mean, these kind of approaches of like having your sample, rotating it and imaging it, as for example in the CT scan, you cannot really use it for proteins because they're super sensitive to electrons. So you cannot, they do not withstand a repetitive exposure to electrons.
Yeah, proteins are not very photogenic, or I guess I should say electrogenic. I mean, you can only take one shot at them and then they disintegrate. So in particular, you cannot take shots at them from multiple angles.
So what you do instead is that you generate multiple copies of those proteins and you put them in a very thin layer of water, and they will adopt random position and orientation, and then you freeze at once. And then what you send electron rays through your sample, and what you obtain is kind of a similar type of measurement. So you have what we call the projection, so a view of the sample under different orientation. So that's good. We kind of have an ingenious solution for that. But the problem is that since they adopted random orientation, you don't know at which angle every measurement comes from.
This is in sharp contrast with computerized tomography scans, for which you know the angles at which the different pictures have been taken.
So that's, let's say, one of the key challenges in cryo-EM. You want to reconstruct, but you need the information on the angle, and you don't have that.
Now, what algorithms could be designed to reconstruct the protein structures based on these extremely noisy and angle-less cryo-EM images? Well, next time we'll discuss a new machine learning algorithm introduced by Laurène and her co-authors to address this specific problem.